just taking a couple steps ahead. But the real thing I wanted to show you guys is just a summary of what we talked about. I'm going to come back to the slide. What I want to illustrate here, this slide is right after the parietal cell stimulation, where we technically actually left off. We left off last Thursday talking about the surgical interventions for such as gastric bypass, lap band, and the root and Y gastric bypass. Now, I'm going to continue on to more the physiology of how we're going to produce hydrochloric acid in our parietal cells. As we talked about the two different types of cells in the stomach, we saw chief cells that produce pepsinogen that became activated in the low pH lumen of the stomach, and that pepsinogen was turned into pepsin, which were our proteases. Our proteases were the ones that would end up digesting proteins into smaller peptides or even down to their amino acids. So that's one aspect of it where we saw the enzyme. Now we need to see how we create the acidic environment of the stomach through the carbonic anhydrous. We'll take a look at that in just a little bit, but what I wanted to do just is to show you, just to do a quick recap of what we talked so far. So in our schematic, you can see we do have the entire alimentary canal, again, consistent with the external environment, in which anything that is ingested by the mouth makes its way through, takes its way through the cephalic phase. The cephalic phase, again, referring to the head. So here we are at the cephalic phase, which starts the whole process of the mechanical breakdown, where we use the mouth to break our food down into smaller pieces. We mash saliva, which contains the digestive enzymes, namely our salivary amylase as well as our lingual lipase. That mashes it right into our food, and now we call it the bolus of food, which starts a process of making its way into the involuntary phase of through the esophagus into the stomach pouch, where the food is now introduced into the acidic environment. We have our pepsin secreted by our chief cells. We have our parietal cells contributing to the pH of 2 in what's called our gastric phase. And in the gastric phase, what we learned last Thursday is that salivary amylase is inactivated due to the acidic environment, but we see lingual lipase being activated in the acidic environment. What we're going to be covering today is everything that's taking place in the intestinal phase, of which we'll talk about the small intestine. Everything from the stomach, from the pylorus to the stomach, we'll make our way through the small intestine. You'll see we're making our way through the large intestine, and then we're going to talk about defecation. And we need to also talk about 3 accessory organs, namely our pancreas, our liver, and then right behind, tucked behind the liver, is our gallbladder. So these are considered accessory organs because, as I mentioned, food doesn't travel through these areas. So anything where food travels through is considered the alimentary canal. These are going to have exocrine functions in that they're going to produce things that will assist in digestion, specifically within the intestinal phase. Okay. And once we make our way back into the small intestine, we're back at a pH of about 6 to 7. Cephalic phase was around pH of 7. By now we have our enzymes, which we discussed, salivary amylase. We have lingual lipase. We have our pepsin. And then we'll talk about the other enzymes that are produced by the pancreas, the production of bile by the liver. We'll talk about a couple more liver functions as well, since we're already on that topic. And then the rest of the intestines we'll cover. With that, now we're going to look at, take a little step back and look at our cells. of which we discussed parietal cells, the cells that are going to produce hydrochloric acid. So what I want to talk about is the mechanism at which we produce HCl, hydrochloric acid. And the main enzyme that's involved in this whole process is the enzyme that we've talked about quite a bit, carbonic anhydrase. So our parietal cells contain lots of this enzyme. Because what's going to happen is, here you can see the lumen of the stomach. So this is our acidic environment. We're going to have that thick layer of mucus that's going to separate the acidic environment with that of the epithelial cells. And then outside of the epithelial cells, we'll have our interstitial fluid, and then we'll have the capillary that runs through. So everything from this point is considered extracellular fluid. And we know that these epithelial cells, as well as the parietal cells, are not damaged by way of that thick layer of mucus that separates the high acidity from our normal pH for these cells to operate. So that carbonic anhydrase, again, our parietal cells that are embedded with these epithelial cells are going to convert through the process of CO2, carbon dioxide, plus water will convert into carbonic acid. And that carbonic acid will dissociate into our proton as well as our bicarbonate. So that process with carbonic anhydrase is the same. So what we need to talk about is, how do we get these components, how do we get CO2 and water into the cell, and through our conversion, how do we produce hydrochloric acid on the apical side of the parietal cell? So let's start off with our parietal cell. Parietal cell is metabolically active, and I'll show you where that step is taking place. And through that metabolically active parietal cell, we're producing carbon dioxide. We use ATP in order to produce carbon dioxide. Carbon dioxide mixes with water through carbonic anhydrase within our parietal cell, will then produce produce carbonic acid. That carbonic acid will dissociate. Our hydrogen ion will make its way through, and then over on the apical side, we're going to see what's called a proton pump, similar to what you guys learned about for our sodium-potassium pump. But the key difference is we're allowing potassium in, but we're shooting out protons. And given that our hydrogen ions are moving from a low concentration of hydrogen ions, okay, so here the concentration is low, into a very acidic environment where we have a very high concentration of hydrogen ions. So we're moving against the gradient. We've got to pump those hydrogen ions out into the lumen. So therefore, we're going to use ATP. This is active transport. And since we're pushing out a proton, we're also going to be pulling in a potassium ion, which actually will make its way right back out through these open channels. So potassium kind of circulates all the way through. Whenever you're pushing a positive ion off, you're going to pull a positive ion in, and you're going to keep the cell electrically neutral. But nonetheless, the main idea is to push protons out into the high concentration of hydrogen ions. So we've pushed that hydrogen out. Now, where do we get that bicarbonate? That bicarbonate that split off the carbonic acid will then shift out into the cell. Will diffuse from a high concentration over to a low concentration. And given that we're moving down the diffusion gradient, we're not using ATP. So here we're going to end up using what's called an antiporter. An antiporter pushes that negative ion out and pulls chloride in from the extracellular environment. So again, from our early lecture in cellular physiology, we mentioned that chloride ions are high in concentration on the outside of the cell. So from the interstitial fluid, it'll get pulled in. While we push bicarbonate out, chloride gets pulled in. And chloride, again, will move down its diffusion gradient down back into the lumen. So I'll touch back on that in just a little bit. Bicarbonate makes its way into the interstitial fluid, into the capillaries, to contribute again to the buffering capacity of our blood, to maintain a pH between 7.35 to 7.45. So that bicarbonate helps buffer any changes in the concentration of hydrogen ions. So if there's metabolic acidosis taking place or any alkalosis, we can prevent any changes to maintain that homeostatic pH in the blood. Now, what we left off with on the apical side of the parietal cell, we had hydrogen ion making its way out, and we had chloride making its way out. So get these two to mix together, you got a positive ion and a negative ion. These opposites will attract, and you get hydrochloric acid. So all the while, that parietal cell is continually churning out hydrogen ions and chloride ions. We're going to contribute to the acidic environment that's taking place in the stomach, and that's how we get a pH of 2. What questions do we have about this?
Where is this taking place?
This is taking place in the stomach. So this is the parietal cell embedded within the mucosa. So just taking a quick step back. Here you see the gastric pits. This is the epithelial layer of the stomach. We have these gastric pits, and deep within the gastric pits, we have our chief cells that produce our pepsinogen, and we have our parietal cells that produce hydrochloric acid. And it's going to make its way all the way up, and it's going to get diffused out into the lumen. Here's our lumen of the stomach, and here is that heavy mucus layer. All right, so now we know how the parietal cell turns on, or at least we know how it produces hydrochloric acid. Let's talk about how it gets activated. And as we mentioned, bless you, as we mentioned, digestion is a parasympathetic activity. Again, parasympathetic pathways, rest and digest. So the postganglionic neurons that innervate the stomach are going to be secreting acetylcholine, not epinephrine. Epinephrine is used for the sympathetic pathways. Acetylcholine is going to be our main neurotransmitter. So we have cholinergic neurons that are present on the basal side of the parietal cell. And as the neurotransmitters of acetylcholine make its way through, it'll come in and activate and turn on our parietal cells. Parietal cells will then undergo the process of creating hydrochloric acid, and that contributes to the low pH of the stomach. So this should make sense. If the parasympathetic pathways, if the sympathetic pathways are activated, we're going to get a bunch of epinephrine that's going to make its way into the stomach, and that won't turn on the production of hydrochloric acid. It's only until we get the parasympathetic pathways that get activated that secrete acetylcholine, bind to the cholinergic neurons, that turn on the process of production of hydrochloric acid. So this should make sense in terms of the digestion ramping up through these pathways, as we illustrated before. That's one mechanism. The other mechanisms involve gastrin, and gastrin were secreted by the G cells we mentioned in our stomach slide. These cells, or these hormones, are hormones that will come in and activate, so the endocrine cells that are embedded within the gastric pits. And let me go back to that earlier slide. We do have G cells that are located that will produce our gastrin. Gastrin will come in and work locally. It's not working through the local bloodstream. It'll work locally to come in and activate those parietal cells. Those parietal cells become activated and therefore again produce hydrochloric acid. The last one of these is histamine. And histamine is a paracrine hormone. And these are actually produced by cells much deeper within. We do have other layers contained within, namely the lamina propria. So if you read through the book, it'll mention histamine. And the lamina propria will then, there's specific cells called mast cells that will secrete this histamine. And histamine, again, a localized response that would also activate these chief cell, the parietal cells to produce hydrochloric acid. So all of these are a local response. So that's really it that we need to talk about in terms of the two cells, namely your chief cells as well as your parietal cells, and then our G cells for the production of our gastrin hormone. The other hormones that we want to talk about are involved in, that have a major control over appetite. So leptin and ghrelin. Leptin, the first one we're going to talk about, is specifically a hormone that's produced by fat cells. Leptin produced by fat cells, ghrelin produced by stomach cells. And they work by giving us sensory information that's led into that will tell us whether we need to increase our appetite or decrease our appetite. And leptin is something that, like I said, is produced by the fat cells for the sole purpose of decreasing your appetite. So this should make sense in a sense where in those that are in obese people typically have more fat cells. So fat cells will produce excess leptin, and that excess leptin should then decrease appetite. So those that are typically thinner, that have less fat cells, will typically have a higher appetite. Okay? And from an evolutionary perspective, that should make sense. If you don't have enough body fat, you're going to have less leptin. And the less leptin, the less capacity you have to decrease your appetite. So in other words, thinner people are hungry. Okay? So in that sense, what that whole purpose of leptin is to let you know, pretty much to govern your level of your appetite. Now, what I want to talk about is just a story back in the 90s when they discovered leptin and ghrelin. They had experiments that they worked on what are called knockout mice. And they take the gene of their standard lab mice, and they found the gene that encodes for leptin. And what they did was they basically come in and destroy that area of the gene so that that mouse can't produce leptin. And what they noticed about these particular mice that didn't make leptin, they were severely obese mice. Okay? And that should make sense, because if they can't produce leptin, they constantly have an appetite. And these mice would just consistently overeat. But what they did was they gave leptin back to these knockout mice. They inject leptin back in there, and they found that these mice all of a sudden started losing weight. They were able to regulate their appetite, and then over the course of a couple weeks, they were back to their normal weights. So Amgen caught wind of this whole study that said leptin has the properties to reduce body fat. And so Amgen bought the rights to leptin. They created their own version of leptin and basically dumped in, I think if this was back in the 90s, they dumped in about $20 million into this whole project. And this leptin would be theirs because they thought they found the secret for bringing weight loss, essentially, bringing down body fat in people. So they, as you know, we go from mouse trials and then we start to bring it out to clinical trials. We had clinical trials as a result. Amgen started their clinical trials on obese people, gave them leptin, and found out technically it really didn't do anything. So this was kind of a $20 million sunk into something. But what they did find was that it wasn't necessarily leptin. Remember, leptin is a hormone, but in order for that hormone to do anything, it needs to bind to a receptor. So in the cases where we see obesity, where it's not the leptin that's an issue that they discovered with our knockout mice, leptin needs to bind to a receptor. And the issue where in cases of morbid obesity, why their appetites were not regulated, even though they had normal and even actually technically excessive levels of leptin, they found that the receptors were constantly bombarded by this leptin. And the receptors no longer could receive leptin. Again, kind of tying into the whole thing of leptin receptor resistance is the term I'm looking for. Very similar to that of insulin resistance, like type 2 diabetes. Type 2 diabetes, you can produce insulin just fine. In fact, you're producing a lot of insulin. It's just the receptors aren't binding as optimally to the receptor. So in order for this leptin to take place, it needs to bind to the receptor. If the receptor is constantly bombarded, it loses its ability to bind to leptin. So the issue that was skirted by the people that studied those knockout mice was that it had nothing to do with leptin itself. It was all due to the leptin receptor. So in these cases, this leptin receptor resistance is basically saying, in the cases of morbid obesity, their fat cells were constantly producing leptin, but they couldn't bind to the receptors to decrease their appetite. Okay? So this kind of ties into the whole thing of, and looking at normal weights, the effect of leptin is to tell you you're full, okay? And that will regulate your appetite at that point. Now, consistently, if you override this process, you'll start producing more and more leptin to try and shut off this process, to decrease your appetite. And therefore, it can come in and start overbinding to these receptors. These receptors create the receptor hormone complex that needs to get degraded on the cell membrane, and therefore, this process isn't optimal for decreasing appetite. So that's the process of leptin. But the other side of it all, we need to talk about what's going to stimulate the appetite, and that's where ghrelin comes in. So ghrelin is produced by the stomach cells. It receives information from stretch receptors, things within the stomach. It's receiving information from the central nervous system, letting us know that the process of appetite stimulation is ramping up. So with that, when we measure ghrelin levels, let's say throughout the day, where would we typically see the highest levels of ghrelin? You would expect high levels of ghrelin right before feeding. Okay? And in fact, they've done studies on those that have performed, that do intermittent fasting. And intermittent fasting, I know I may have just talked about prior, but intermittent fasting is a period of about 15 to 20 hours of fasting. And they found that ghrelin levels are very high right before feeding. And that should make sense. Ghrelin levels are high, you've got a very strong stimulant letting you know, bring up your appetite, you're about to eat. Now, as we start to fill up our stomach, so the chemoreceptors are now picking up the changes in the stomach. The stomach is starting to fill up, and the stretch receptors are now coming in and also feeding the central nervous system, letting us know that our stomach is filling up. And that will do the exact opposite, where it'll come in and inhibit the process or the production of ghrelin to reduce your appetite at that point. So looking at these two, leptin and ghrelin, basically are antagonistic hormones. They work one against each other. One will decrease the appetite, while the other will stimulate the appetite. And I don't believe the book goes too much into this. This is something that I wanted to kind of throw in, because these are key hormones that were discovered, I wouldn't say recently, but within the 90s and 2000s. The next slide really just goes over looking at the two, the ghrelin, the appetite stimulator, and leptin, the appetite suppressor. And they work hand in hand. Leptin, which is going to decrease your appetite, again, taking place after you eat. Ghrelin will be the highest right before you eat. And they work hand in hand in order to stimulate your need to feed. Okay, what questions do we have about those two hormones? All right, we've talked about pepsin so far. The other one I forgot and neglected to mention is gastric lipase. And we'll continue on with the rest of the intestinal phase. And first, what I'd like to talk about is our pancreas. So we've talked about our liver and our pancreas as our accessory organs. Let's take a look at the pancreas. Now the pancreas, which you can see, is embedded right below the liver, nestled up right against the duodenum of the small intestine, which we'll talk about when we cover the small intestine. The key thing about the pancreas is that it produces our enzymes. We've got a couple pancreatic enzymes to talk about, and then the presence of high amounts of sodium bicarbonate. And that aims to buffer the chyme that makes its way from the pylorus of the stomach. Here's our pyloric sphincter. And we're going to dump all this acidic chyme into the small intestine. So the pancreas is producing that buffer that comes in, that bicarbonate that comes in to neutralize the acidity of that chyme to introduce us back to a neutral pH. So here you can see the production of those, again, our enzymes and our bicarbonate, and we're going to continue on with those enzymes. We're going to continue on with digestion in the small intestine. More to come on that. The other portion that we want to talk about the pancreas is that the output, we'll look at, in the next couple slides, we'll look at the functional units within the pancreas for the exocrine functions. And we're going to see how those enzymes as well as our sodium bicarbonate are going to make their way out and to the primary output. And that primary output is called the hepatopancreatic ampulla, or the ampulla of Vater. And why is it called the hepatopancreatic? Hepato meaning liver. Here you'll see the liver for its pathways for producing bile. It shares the same output as that of the pancreas. And you get this little nipple right there that's embedded within the mucosa. And the purpose of that is to basically dump out the contents of the enzymes, as well as bile produced by the liver, as well as the sodium bicarbonate produced by the pancreas. You also do have other outputs called the accessory duct, so the duct of Santorini, but the main output would be that hepatopancreatic ampulla. And where this takes place, it's about 4 inches. Here you'll see the length of the duodenum is about 10 inches. The first 4 inches are going to be the point at which we're going to dump these. So to kind of tie in back to what we talked about last Thursday, when we saw the root and wide gastric bypass. There was that question that I sort of asked of why, with the presence of the stomach, you saw the esophagus into the stomach, and from the stomach pouch, the remnants of that stomach was still attached to the duodenum. And you saw that we had an anastomosis that attached that little pouch, that little stomach pouch, and connected all the way to the jejunum. What was the sole purpose of this duodenum if we're completely bypassing it? The whole purpose of that is that the pancreas and the liver still need to do their job. And again, to produce the bile, the enzymes, as well as the sodium bicarbonate that are going to get dumped into this region and make its way down to the jejunum where we had that other attachment. So we don't want to get rid of this, because if we sever that duodenum, we have no way of using the enzymes that are produced by the pancreas as well as the bile for that continued digestion. So any way you look at it, this is where that action takes place. So let's take a step back and look at how we produce the stuff that the pancreas produces. We're going to backtrack to what we looked at before when we looked at our acini. So the pancreatic acini are going to be the exocrine cells. We're going to zoom in for the pancreas. We're going to zoom in into this little cluster of grapes at the very ends of each of these pancreatic ducts. And within these, we'll zoom in even further, and you can see our cuboidal cells. You can see all of our endocrine cells, of which we have the alpha cell, the beta cell, and our delta cells, all embedded. So this is our endocrine. And the acini on the outside, in yellow, are going to be our exocrine cells. And it's these cells that we want to focus on now, because these are the ones that are going to be producing the pancreatic juices, which are going to contain the enzymes as well as the sodium bicarbonate. So these cuboidal cells are tasked to produce those. They do their production. Here you can even zoom in a little bit further. You can see the production that will dump into the ducts and therefore make their way over to the hepatopancreatic ampulla. Now, looking at the other endocrine cells, just as a recap, you can see our alpha cells, which are going to produce our glucagon, our beta cells, which are going to produce the insulin, and the delta cells that are going to produce our somatostatin. And where did we see somatostatin? We saw that in our endocrine lecture. Somatostatin was our growth hormone inhibiting hormone. So the purpose of this growth hormone inhibiting hormone, remember our growth hormone was responsible for the breakdown of carbohydrates and fats while preserving proteins. So in the case of somatostatin, somatostatin produced by the delta cells will come in and turn off the production of growth hormone. And that comes back over to our hypothalamus, to the pituitary, to prevent the production of those. So that's where we get the inhibition of growth hormone production. Looking at the makeup of the pancreatic juice, as I mentioned, enzymes plus sodium bicarbonate. How much of it do we produce a day? Is about 1.5 liters of alkaline pancreatic juice secreted right into the duodenum by way of the hepatopancreatic ampulla. So just to sum up the total volume of our juices, we had 1.5 liters a day for saliva. We had 3 liters for that of gastric juices. Now we have another 1.5 liters in pancreatic juices. So by now, we have 6 liters total of fluid that's dumped into the alimentary canal. And obviously we're going to have to reclaim that water. And where is that going to take place? That'll take place in the small intestine, but I'll talk about that when we get to that section. So the whole purpose of the pancreatic juice is to, again, buffer that pH. We have the acidic chyme that's making its way into the small intestine. We want to be able to buffer it, and that's where we get our sodium bicarbonate. Or rather, that's the whole purpose of the sodium bicarbonate. The other enzymes, you'll see, we have pancreatic amylase. Again, amylase for the purposes of digesting starches. We have our polypeptide enzymes. So if this is our starch, or these are our carbohydrates, our peptides, our polypeptide enzymes for proteins, and the last, pancreatic lipase for the fats. Looking at these, you'll see 5 polypeptide enzymes. I know you only see 4 on there, but the 5th one, which I'm not really ever ask you, is called enterokinase, or also known as enteropeptidase. So there's actually 5 enzymes: trypsin, chymotrypsin, carboxypeptidase, elastase, and then lastly enterokinase. So the acronym here that they use in medical school is ETCCE for eat, tender, chicken, chunks, elegantly. I know these are silly, but that's how you remember these things. So these ETCCE is what's used, and that helps you remember elastase, trypsin, chymotrypsin, carboxypeptidase, as well as enterokinase. So all these, and I won't really go into the real main differences of all these. The main four right here, the whole purpose of this is really just to digest your proteins. You have long proteins from the food you've just consumed, and we want to be able to break that down through catabolic reactions into smaller peptides. Those smaller peptides, once we break them down into the smaller peptides, then down as that food travels down the small intestine, we're able to take those small peptides, absorb them through into our bloodstream, and then construct bigger proteins from that. So we want to create small peptides as well as the amino acids that come from it. So these help cleave these long proteins. They look specifically for a primary sequence of amino acids, and they'll just cut. That's all they do. And they continually cut until you have smaller fragments. Now, technically what's happening is we actually have that enterokinase or enteropeptidase. One of the things that I won't really go too much into is that trypsin is not actually released as trypsin. It's actually released as trypsinogen. Just like you saw in the stomach for pepsinogen, just like you saw angiotensinogen in our endocrine system. Enterokinase comes in and finds trypsinogen and cleaves off a bunch of proteins, leaving you with a very small enzyme called trypsin. So this is where these come in to cleave and turn it into trypsin. And trypsin is now activated to help cleave your proteins. That's really it with the pancreas. The pancreas doesn't really do anything else for the purposes of digestion other than produce all of these enzymes. The other enzyme that we mentioned is pancreatic lipase. And again, looking at the term lipase, breakdown of fats. So we have now lingual lipase activated in the stomach along with gastric lipase, and now we have pancreatic lipase to help create fatty acids and break apart those ester linkages. Now the liver is the other organ that's going to be involved in the production of bile, and I'll talk about the production of bile for digestion, but since we're already here, we'll also talk about the other functions of the liver for the breakdown of red blood cells. You'll see that it's the largest gland, but it's the second largest organ. Do you guys know the first largest organ? The skin, yeah. So liver is right there, right as the second largest organ. We have two main lobes, which we won't really get into. But if you're going to take a look at the input and output of the blood, for the purposes of the liver's filtration of the blood, we've got to take a look at the input as well as the output. The first of the input come in from the portal vein. And this portal vein is a light purple. And it's a light purple because it is actually a bit of a mix of oxygenated blood with deoxygenated blood. And it's coming in from the veins and capillaries that surround the small intestine. So this portal vein that's coming in is going to have nutrient-rich blood, but deoxygenated blood. Now the hepatic artery, which comes off the descending aorta, the hepatic artery will feed in nutrient-poor blood, or nutrient-poor but oxygenated blood. So 75% of the blood that feeds into the liver comes in from the portal vein, the remaining 25% from the hepatic artery. So these feed into, and you can see the output, they basically will spiral together and feed right into the functional units. So we'll take a look at the functional units. The output of the blood that comes in from all of our input, once it bypasses our functional units, will then converge into the hepatic vein. You've got the central vein system that will all verge together into the hepatic vein. Hepatic vein will then dump into the inferior vena cava, returning us back to the right atrium, making its way into the pulmonary circuit. So knowing that we now have two inputs for the blood, we can see where that blood makes its way. So as I mentioned, portal vein and hepatic artery branch off into a bunch of smaller arteries, arterioles, into capillary beds. And within those capillary beds, there's going to be embedded within our functional unit called liver lobules. These liver lobules are a bunch of repeating hexagonal units, and you can see that on our image on the top right. A bunch of hexagons embedded, where right in the very center, you're going to see what's called the central vein. And the central vein is going to be something where it's going to be the drainage into the output. So this represents our output of the heart. Output again being on its way to the hepatic vein. You get these central veins that are going to be collecting blood. Let me first kind of clarify what I'm talking about here. Our portal vein, you can see, is our input of the heart. Input of the liver, I mean. Input for the portal vein, we get 75% of the blood through the portal vein. We get 25% of the blood through the portal arteriole. And they come together and are going to be all present at each of the single points on that hexagon shape. So each of those 6 points in the hexagon are going to consist of what's called the triad. And the triad, if you read about the triad, will tell you about you can see on our bottom right, our portal venule, our portal arteriole, again, branches of our input of blood, and then the last will be our bile duct for the purposes of producing bile. So these three are typically together on each of the sides of that hexagon. And the flow of the blood flows in from the portal venule as well as the portal arteriole, will flow into these sinusoid capillaries, which I'll describe in just a bit, and they'll drain into the central vein, as you can see right in the very center. So you see this point at which you get a bunch of spirals that are making their way in. They're all converging onto that central vein that will jump in and join these interlobular veins, which will all dump into the hepatic vein back to the inferior vena cava. So you can really see how the liver lobules serve to act as a filter. Everything from this, our triad, over down to the central vein. So what's taking place right in between? Again, we're going to have blood that's flowing through, and then we have the production of bile, which we'll see in just a bit. Now let's zoom in, and here you can see our triad again, our hepatic artery. You'll see that this was our portal vein, and then our bile duct. So here you can see the venous blood that makes its way in. They're going to now converge into these sinusoids, and then as they make their way through the sinusoid capillaries, they'll drain back into the central vein. So what's taking place in between are each of these liver cells, and these liver cells are called hepatocytes. Hepatocytes. Oh, it's right there labeled off to the right. Hepatocytes are going to be these liver cells that are going to have numerous versatile functions. So looking at this pathway of blood, you're making its way through. You've got these capillaries, and you've got all these hepatocytes that are going to come in and assist in the breakdown of stuff that's present within the blood. So there's this definite filtration as it makes its way through. And then once you've cleaned up your blood, you're going to drain back into the central vein to return that back over to the inferior vena cava. So this should make sense where anything that you've just consumed, anything that, whatever makes its way into the small intestines, gets absorbed through, we want to be able to come in and remove things that shouldn't be present in the blood. So why the sinusoid capillaries? I'll point out in a later slide. All right, so let's talk about the hepatocytes. Each of these individual cells, these red dots right here, what purposes do they have in terms of filtration? We have two functions we need to talk about: digestion and then non-digestion. digestive. The digestive purpose is for the synthesis, transformation, and storage of protein, carbohydrates, and fats. Again, blood coming in from the portal vein from the small intestine. Here the liver can come in and store things that we might have in excess. So if you've just taken in a bunch of glucose, that glucose is going to make its way into the portal vein. It's going to raise blood sugar. And what better way to store that excess glucose within the liver? And that storage process, we mentioned, we want to create liver glycogen. And how we produce glycogen is through the process of glycogenesis. This is also where we get the process of gluconeogenesis, where any of that lactic acid that makes its way through, the liver can convert any of those back into available glucose. And if that glucose will be returning back into the bloodstream, it'll make its way into the central vein. Otherwise, if we want to store that glucose, we can store it as glycogen within these hepatocytes. Not only can we store glycogen, we can store proteins, and we can also store fats as well. Take a look at why this is important. Why do we store fats on there? This is another area, aside from fat cells, adipocytes, that we can actually store fat in. Has anybody here ever had, I'm probably butchering the name of it, foie gras? Yeah. Fatty duck liver? It's kind of a fancy food that I've never personally eaten. I don't think I'd ever eat it, but typically what they would do is they would take geese and they would feed them very high-fat diets. And because they have such a high-fat diet, they end up storing a lot of that fat in the liver. And they take that liver, they crush it up, create like a pate out of it, and then serve it. I'm too rich for my blood. I've never eaten it, but nonetheless, that's the whole purpose of it. Take something in the case of you have a fatty diet, and you get a fatty liver. Okay, so that's what we do with it. The other purposes of the liver is detoxification. What are we detoxing from? Drugs and alcohol. Anything that's taken in that makes its way into our circulatory system needs to get broken down. And that's exactly what the liver is supposed to do. Now, abuse that system, and your liver is constantly working overtime, and will start to break down over time, and you're going to see what's called something called cirrhosis that you'll be able to explore on a later slide. We can also modify some of the molecules that are taken in, and then we can excrete anything that we don't need. The non-digestive is the phagocytosis of old or worn out cells. What worn out cells are we talking about mainly? Our red blood cells. Our red blood cells, as you remember, have about 100 to 120 day life cycle. They don't have mitochondria, they don't have any nuclei, so they can't regenerate their own cells, they can't repair themselves. After about 100, 120 days, they live out their life cycle, and we're not going to just, dump out red blood cells out of our body. We're going to want to break that down. So where you saw the sinusoid capillaries in the bone marrow, where we saw the production of red blood cells, you're going to see the same sinusoid capillaries present within the liver lobule for the purposes of taking that red blood cell, breaking it apart, and reusing that for the new production of red blood cells. Okay? So we're going to break those old red blood cells down. Let's see if I got an old slide for that. Nope. Okay. The other things that the liver does, also produces a lot of our major proteins. So the major proteins that are present in the blood plasma, things that are dissolved in the blood plasma: heparin, prothrombin, fibrinogen. These mean three things: blood clotting. Albumin, you've seen albumin before. That was our lipid-soluble hormone carrier. And then lastly, of all of these, you've definitely seen angiotensinogen. Angiotensinogen was the protein that's present in our plasma that in the process of the RAS system, where we get either dehydration or low blood pressure, angiotensinogen is converted into angiotensin by way of renin. Renin produced by our kidneys. So it'll activate it and turn it into angiotensin, and angiotensin will go through and get converted into angiotensin 1 to angiotensin 2, angiotensin 2 to aldosterone for raising blood pressure back up. So these are our non-digestive proteins that the liver produces. The other main thing that we'll talk about on a slide later is the production of bile. And I'll talk about that on the later slide. Here you can see again the pathway of blood, where we saw 75% from the portal vein, 25% from the hepatic artery, makes its way into the liver. This makes its way to the triad, into the liver sinusoids. Sinusoids then drain back into the central vein. So here you see these sinusoids all within. These are the little passages in between. You see the hepatocytes embedded right around the sinusoid capillaries. The sinusoids then converge onto the central vein, where from the central vein they converge into the hepatic vein. Hepatic vein is the one large vein that dumps right into the inferior vena cava. And that makes its way back into the right atrium to continue on with the pulmonary circuit. The other thing I want to look at are the other cells besides the hepatocytes. We also have fixed macrophages called Kupffer cells. And Kupffer cells, for their purpose is just basically to filter out any red blood cells, white cells, and any bacteria within the blood. So macrophages help basically clean up that blood. The other function that we need to talk about is the production of bile. And these hepatocytes, and keep in mind, I did mention a lot of different hepatocytes, or I'm sorry, a lot of functions of the hepatocytes. The functions of the hepatocytes really differ based on what thing they're next to. So the hepatocytes that are right next to what are called the bile canaliculi are the ones that are going to be producing bile. And bile is another exocrine product that help emulsify fats. And I'll talk about that on the next slide over. The emulsification of fats is our way of basically mixing fats into water. As that bile gets dumped into the duodenum, this is our way of our absorption of fats, and that's where bile is absolutely necessary. Now, how much do we produce in terms of bile? Now we have 1 liter of bile per day. So our running total for the amount of water that we've dumped into the system is now 7 liters. 1.5 in saliva, 3 in gastric juices, 1.5 in pancreatic juices. Now 1 liter of bile. That's 7 liters now. Per day. Now, what exactly does bile do? Well, bile, like I said, emulsifies fats. And the way it works, it has both hydrophilic and hydrophobic regions. And it comes in and circles around a lipid molecule. So here's our lipid molecule. It's all clumped together because this is a rather large molecule. And bile salts, typically they're called bile salts because they're actually like, they're similar to phospholipids. But they come in and will take that lipid and break it apart. And that breaking apart takes these little lipid droplets and encapsulates them in bile. So what exactly is the process of emulsification? If I can help you understand what emulsification is. Emulsification is taking fats and being able to dissolve that in water. So if I took oil and I took water and I put them together, they'll separate. Obviously you'll get the fat on top, you'll get the water separately. And that's not really going to be useful. Imagine that making its way into the small intestine. We have no way of taking those lipids, which are valuable. we have no way of absorbing that huge chunk of lipid. All those fats are stuck together. So what we need to do is to cleave all those lipids that are all stuck together and make them into smaller droplets. Those smaller droplets allow for increased surface area so that we can individually absorb the bile as well as those droplets. And we want to introduce that back into the circulatory system for use later on so that we can store those fats. So what exactly is the process of emulsification? Let me give you the example of an egg. And if you crack open a raw egg, you'll see the egg white and you'll see the yolk. And those two are typically maintained separately. It's because the egg white is mostly albumin, it's water soluble, but the yolk is going to contain a bunch of cholesterol, it's going to contain a bunch of fats in there, and those two don't mix. How do you mix them together? Well, you get a whisk and you mix it really well. And then you notice now it's all kind of mixed together, but technically they're not really mixed, they're just kind of embedded within each other. If you left that solution to sit for a little while, you'll notice that they'll start to separate after a little while. Another thing is milk. If you ever look at milk, milk is a combination of water as well as proteins that are dissolved in that water, but there's also fats in there. So if you ever look at whole milk or 2%, maybe not skim milk, but if you look at milk, milk is an emulsion. You've got fats dissolved in water. Let that milk sit out for a while, and you see a clear separation of fats on top of the water. So in this process, we're doing exactly that. We want to take those droplets of fat and mix them into the watery aqueous environment that it's going to make its way into once we're in the small intestine so that we can absorb this stuff, because this is important. There's tons of calories in our lipid that we can use to make our way back into the digestive system. Now, without these bile salts, if you have less bile, and why would you have less bile? This would be in the case of anybody that's ever had their gallbladder removed. So that is, which we'll call later as a cholecystectomy, is the removal of the gallbladder, which is supposed to concentrate bile. And I'll talk about that concentration of bile in just a bit. So in those cases where people have had a gallbladder removed, surgically removed, they have less ability to absorb fats. So typically people that have had their gallbladder removed will be advised to modify their diet, to not take in as much fats. And why is that? Because you have no way of emulsifying the fats. They'll make their way into the digestive system. You've got a bunch of lipids that are stuck together without that excess of bile, and that could cause some gastrointestinal distress within the large intestine, all due to the bacteria present that cannot react to a bunch of lipids all stuck together. Now, the other thing about bile is that bile, the dark pigmentation that comes from bile is called bilirubin, and that comes from the breakdown of these old red blood cells within those hepatocytes. So the hepatocytes over here can come in and you have these sinusoids that come through. We can take in those red blood cells and break them apart. And that breaking apart, that byproduct is called bilirubin, and it comes from breaking apart those iron particles within hemoglobin. So that bilirubin, which you'll learn later on, is what contributes to that brown color of feces. Most of that, where that color comes from, why is your feces brown? It's bilirubin. The thing of why you're seeing this image to the bottom right, has anybody ever seen this before? For Lay's Light? Still out on the shelves, you don't see too much of it, but this was something that was heavy around the 90s and early 2000s. It was a product where they were able to find a so-called fat substitute, but it wasn't so much a substitute. It was called Olestra. And Olestra was a molecule that came in that also had the ability to interfere with bile salts. And the theory behind it was, and I've been describing the whole process of taking lipids and breaking them apart into these smaller particles that can be absorbed. But what if, in the purposes of diet modification, what if you don't want to absorb those lipids? You get this molecule called olestra that interferes with the binding, and now these lipids don't get absorbed. And eventually it'll make its way through the alimentary canal, and you can eliminate a good amount of fats without actually absorbing it. So this company created olestra for that purpose. And they started to introduce it to foods as a way to reduce the amount of fat that you absorb. Now that you don't have this emulsification, what happens in a lot of these cases after they've released this product is that some of the things that they found about people that ate a lot of this stuff is that fats are extremely important, not because they contain calories, but because there's also fat-soluble vitamins. Namely vitamin A, D, vitamin E. They're all fat-soluble, and they need fat in order to dissolve so that you can get that into your system. Now, if you can't absorb that fat, you don't get those vitamins that you need. And the other thing that's also involved is that those lipids that make their way through the large intestine cause a bunch of gastrointestinal distress. And one of the common side effects of people that ate these particular olestra-containing products had what was a condition called steatorrhea. And it kind of looks like the term diarrhea.
Fat in the excrete fat in your.
Extreme, yeah.
There's a certain fish that does that too, like whitefish. That if you eat too much of it, you can have that.
Really? What
Yeah.
kind of fish is that?
It's whitefish. If it's too much whitefish, it's like too much fatty fish.
Oh, okay.
My dad had it, so I knew about it.
Interesting. All right. Well, thank you for sharing. That's basically what Nancy was describing. Is the easier term for this is called anal leakage. It's the inability to allow fats. Fats would end up leaking and separating from the feces, and will end up causing what's called anal leakage. And what's that?
They mentioned it in a movie.
Through olestra?
They mentioned the anal leakage from chips. He's like, don't you know those chips give you anal leakage? I was like, what? It's on the warning label.
Yeah. So the warning label, which has like a fine print in the very back that you don't read.
That's so funny.
Yeah. So that was something common, and they found people that were staining their underwear. And I won't really go into too many of the details, but it was just something common. So they didn't quite pull it on the shelves. I know I've seen them around. I remember seeing at the very bottom of the chips aisle and saying, damn, they still sell olestra. And it's there.
That happens with Ally too, the diet pill or whatever.
What was it called?
Ally,
Ally? How do you spell that?
ALLI.
Okay.
I had a coworker that used it, and she said when she went to the bathroom, you could see the fat.
Oh yeah, is that the dieting pill?
Yeah.
My friend did that. She told me about it. I was kind of weirded out. Like,
She lost like 100 pounds, which she gained back. But she was like, when every time she went to poop, anything, because you were supposed to have a high fat diet, she's like, it all came out.
Oh, gross.
Yeah. It's like,
So, yeah, avoid these products. For the sake of yourself and for the sake of your underwear.
is it here?
That's what's funny is I remember when I was taking physiology and I didn't learn about olestra until I started reading more and more and started putting these slides together, and I'm like, I remember eating these olestra chips, and I remember, and I started reading more about it. It was one of those, that's why that happened. I could never really explain it, but thank you,
it's like the
physiology.
sugar-free gummy berries.
Sugar-free gummy bears?
The Haribo sugar-free gummy bears give you some sort of like awful diarrhea too, like explosive diarrhea. If you go on Amazon, you can read the comments of people that just, explosive diarrhea.
Wow. And I think people take also Amazon pictures too. I've seen. Anyway, got it. So stay away. So, modification of your diet really should be taking place prior to introducing it into the alimentary canal, as opposed to interfering within the system. Because as we've kind of been able to illustrate, anything that comes in and changes the process, especially of that of fat absorption, can really cause some major changes that we don't want to happen. So, as I mentioned, the bile. So now we're on the production of bile. We know that the liver has all these other functions, but we want to focus on the digestive portion. The bile canaliculi, which are present right in between the hepatocytes, come in and start dumping that bile that's produced into these hepatic ducts. You've got the left hepatic duct and the right hepatic duct, which all converge together into what's called the common hepatic duct. And the common hepatic duct then binds to or not binds, but joins that of that called the cystic duct. So gallbladder that's off to the bottom of the liver, right behind the liver, will come together. So cystic duct and the common hepatic duct come together as what's called the common bile duct. And common bile duct then merges with that of the pancreatic duct to create what's called the hepatopancreatic ampulla. So with that, we see the process of where bile is dumped into the duodenum. Let's talk about how, and here you can see how those come together. Our common bile duct as well as our pancreatic duct. Here you can see the hepatopancreatic ampulla that comes in and merges right into the small intestine, penetrates right through that mucosa in order to dump the pancreatic juices, the sodium bicarbonate, as well as the bile right into the small intestine. What's also present is this sphincter. So you do have this muscle layer, the mucosal, the muscularis layer is present with this smooth muscle that wraps around that particular ampulla in order to regulate the release of bile. So in this process, we're not constantly releasing bile into the small intestine. We do have to regulate it. So in the cases where in between meals, where we don't have food passing through, that sphincter is typically closed up. And by closing it up, that's going to now close off or occlude that ampulla, which will cause backup of this bile all the way through. So that backup will make its way all the way back up. And once it backs up, you'll have where the gallbladder comes in. You'll get the bile that backs up all up into the gallbladder. So we've got this watery bile that makes its way into the gallbladder, and the gallbladder will help in concentrating that bile by removing the water as well as the ions that are present within that bile. So now we get this concentrated bile that backs up, and what happens as food travels down, food makes its way into the stomach. The stomach now is going to start feeding information back into the central nervous system. You're going to have hormones traveling through, and the hormone that's going to help release the gallbladder, cause the gallbladder to contract, is CCK, and I'll show you that in our next slide over. So here you can see the back of the liver. You can see the gallbladder. CCK, cholecystokinin, is what's going to cause the release, and I talked about this a little bit in our stomach slide, where CCK comes in to contract the gallbladder. So why is it called cholecystokinin? If you break down the word, cysto means gland, chole means bile, and kinin, like kinesiology, is to move. So bile gland move is basically the name of it. So this hormone comes in and causes that gallbladder to contract and to secrete that concentrated bile to make its way down the common bile duct to dump right into the hepatopancreatic ampulla. So in those cases where if you've ever had a gallbladder removed, is what's called a cholecystectomy. Again, looking at chole for bile, cysto for gland, tectomy, which is the removal of. And typically, as I mentioned earlier, typically a modified diet, a diet lower in fat due to the lack of concentrated bile within the gallbladder. Now, even though you have a lack or less bile, you still have other lipases involved. Again, lingual lipase, gastric lipase, as well as pancreatic lipase. And if you go back into your homework, you don't have to bring it out right now, but your homework will look into summating all of these enzymes that we've talked about so far. So that way, that serves as a good table for you to kind of help summarize your notes and then piece it together in preparation for the final, at least for the new content within the final. All right, let us take a little break. You guys want to take a little break before we finish? All right, how long would you guys like? 10 minutes, 5, 10, 5, 10? 7.